All right, we're right on time. So, hello everyone. Welcome to our webinar, Next Generation Cell Therapy Innovation, Personalization, and Clinical Expansion. My name is Peter Xue. I'm the Marketing Product Manager at Acrobio System. It's a pleasure of mine to host today's discussions. As the field of cell therapy continues to evolve, we're seeing new opportunity emerging from NK cell and gamma delta T cell. These immune cells are the forefront of innovations, really offer fresh possibility for improving outcomes in cancer and other diseases. Over the next hour, you'll hear from leading experts talk about their research and walk us through some of the latest advances. With a fundamental biology to clinical applications, we're excited to bring together perspectives that help us better understand where the field is headed. All right, without further ado, I'm like to introduce the second speaker. Next, I'm delighted to introduce Dr. Kate Rockland. She's the Chief Operating Officer at Innate Bio, a clinical stage company advancing gamma delta T cell therapy in oncology and autoimmune diseases. With more than 20 years of experience in company development, operation, and clinical manufacture advanced therapies, uh, Kay has held a leadership role across biotech, including serving as Chief Business Officer at Curadigm. She earned her PhD in molecular biology and genetics from Cornell Medical College and conducting research at Sloan Kettering Institute. Let's welcome Kate.
Okay, can you hear me?
Yep, we can hear you.
Perfect.
So,
Let me just do this here. There we go. So my name is Kate Rockland. I'm the Chief Operating Officer at Innate Bio, and we are a gamma delta T cell company. So today I'm going to be talking a little bit about our work in gamma delta T cells. I'm going to try to talk a little bit more generally about how we think about using rare cell types, so both NK cells and gamma delta T cells, and how we think about harnessing the unique properties of these cells, I'll say, to develop some next generation cell therapies. So I think you're going to see some really interesting similarities in sort of the approach that Neil and I are both taking and how we think about our cells. So here's my required disclaimers. And so I sort of want to step back because I'm going to talk a lot about gamma delta T cells, but really I want to sort of broadly talk about what do we think about cell therapy? Where has the field been and where do we need to go to tackle some of the problems that remain? And so there are really three components that I always think of that I think are really critical for cell therapy. One is durability of response, and we talk about durability, a lot in cell therapy. And so duration can be the actual duration, so the time that you see the response in the patient or the cells remaining or expanding or proliferating over time. But durability can also be the depth of response. And so, what I'm actually going to talk about, and Neil touched on as well, is how do we use cell therapy in synergistic combinations with the idea that we need to pull cell therapy in some ways out of those late line therapies and pull it more into the frontline therapy. We need to attack these tumors early on with everything we've got. And I think that there are a lot of powerful ways that we combine cell therapies with other technologies to drive those deep responses in cancers, particularly difficult to treat cancers. Cells also need to be tolerable. And so I think, from both an NK and a gamma delta T cell perspective, we've seen a lot of challenges with traditional alpha beta T cell therapies. So we've seen some toxicities, we've seen some issues there, but most importantly, a lot of cell therapies worked early on, and I sort of joke sometimes that we found the needle in the haystack first. We found the CD19, we found the BCMA CARs, and those work extremely well. But the CD19 CARs don't work because they're a precision medicine in the sense that they can differentiate between healthy and sick tissue. They work very well because they can ablate the entire B cell compartment, and you can survive without that compartment. So when we say tolerability, it's both side effects and toxicities. But as we move into the solid tumor setting, how do we start to think about using cell therapy less like a sledgehammer and more like a scalpel so that we can remove that cancer tissue, but leave that healthy tissue intact? And finally, how do we get at the question of heterogeneity? And again, in solid tumors, this becomes extremely challenging. we know that tumors are not one single disease. They're often many, many different tumor cells. The heterogeneity of these difficult to treat cancers can be extremely high. And so, how can we use some of the endogenous immune mechanisms in things like gamma deltas, NKs, and others to try to cover that tumor heterogeneity without having to over engineer? One of the challenges that we've seen is early on, some cell therapy companies tried to do multiple edits or multiple targets on a single cell therapy. And that can be challenging both from controlling your costs, but also there is a point where we think you start to break into forcing a cell to do something it doesn't naturally want to do or overriding some of those endogenous mechanisms. So I'm going to talk today a lot about how do we lean into the capabilities and the mechanisms of your cell type and leverage that so that it can be the most powerful immune cell it can be without trying to force it into a position where it's unhappy doing something it doesn't want to. So I'm going to talk to you a little bit about gamma delta T cells, which we are very passionate about at Innate Bio, but tell you a little bit about why we think they're such powerful immune cells for cancer elimination and immune surveillance. So gamma delta T cells are really unique cells. They actually sit similar to NKs at the nexus of the innate and the adaptive immune system. And as such, they actually have properties of both. So they can kill directly, very similar to an NK cell, but they can also antigen present and immune amplify responses and recruit immune responses very similar to the adaptive immune system. They actually do not they're not MHC restricted, so they can go from a donor to a recipient without any editing. But importantly, their actual role in the body is to differentiate between sick self and healthy self. And they do this through a very complex set of receptors that allows them to do normality sensing and also sensing of cellular stress. So they naturally sort of surveil in your body and they'll eliminate any cells that may be potentially going down those oncogenic pathways, cells that are damaged, things like UV damaged cells. And so that's really their function. And so they do that through the gamma delta TCR. And that receptor, in and of itself, is a polyclonal receptor. And that receptor can recognize 8 to 10 different stress ligands that we know of. And so it can recognize stress from either DNA damage, it can recognize stress from rapidly dividing membranes. And through this normality and stress signaling, it will essentially make a judgment about whether a cell is healthy and should remain, or if it's sick or diseased and should be eliminated. And so it's been referred to as nature's CAR T, and we think the gamma delta TCR is incredibly powerful in how it actually works to detect damage. So I'm going to tell you a little bit today about how we've tried to leverage that to actually identify and eradicate residual tumor cells. So very early on, the observation was made that gamma delta T cells are associated with greater cancer survival in patients. And a lot of this work actually comes from Dr. Larry Lamb, who's the CSO at Innate Bio. And so I want to show you a little of this. And so on the left here, you can actually see his initial work. And so he published this study. It was originally done as a retrospective analysis in the late 90s, early 2000s. And at the time, he was looking at leukemia patients post stem cell transplant. And what he saw was there was this really significant separation of the curve based on those who naturally reconstituted high levels of gamma delta T cells versus those who reconstituted low levels of gamma delta T cells. And this is one of the first observations, and many groups have made similar sense, that demonstrated that high levels of gamma delta T cells naturally occur in patients that have improved overall survival or improved clinical outcomes. So that's in leukemia. But on the right there, I'm actually showing you a pan-cancer analysis, and this is looking across 25 different solid tumor cancer types, as well as brain tumors and non-brain tumors. And what you can see is they're looking at different components that are either favorably or adversely correlated with clinical outcomes. And the very top there is gamma delta T cells. So these high levels of gamma delta T cells seem to be the key, or at least linked to improve clinical outcomes across multiple different cancer types. And so we took this observation, and this really formed the basis for Innate Bio and the gamma delta T cell platforms that we've developed in-house. So in INB 100 here, I'm actually going to talk to you today about our GBM program, but I wanted to call out INB 100 here because this is actually a clinical program that's based on that initial observation I just showed you. So Dr. Lamb realized in the early 2000s that high gamma delta T cell levels were correlated with favorable outcomes in transplant. We now have a clinical program where we actually give gamma delta T cells in combination with transplant and complex adult AML, and that's our INB 100 program. So it's been very exciting to be part of seeing something translate from an observation 25 to 30 years ago in the clinic into an advanced therapy that's actually completing phase 1 enrollment. We have a number of programs that are in development, but what I'm going to talk to you about today is actually our solid tumor program. And this is our INB 200 and 400 program. And this is in glioblastoma, so one of the most challenging treatment-resistant cancers out there. This is in frontline, newly diagnosed glioblastoma, and I'm going to share a lot of the clinical data with you today where we've looked at some long-term outcomes in these patients that we've treated. So I'm going to go back to the comment that Neil made earlier, which is defeating solid tumors is hard. Solid tumors are incredibly challenging for all of the reasons that he laid out. And I always show this picture because a picture is sort of worth 1,000 words. So here you can see this solid tumor with all the purple staining. And the fact is, you not only have the bulk of this tumor here, but near the periphery or the tumor margins, you can still see a number of these tumor cells spreading beyond where the tumor bulk is. And so not only do you have to overcome the immune barriers in the tumor suppressive microenvironment and the trafficking into the solid tumor, but you have to overcome the fact that there's often these small bits of solid tumor that are breaking away from the main tumor or stratified away in different layers of tissue from where that main tumor bulk is. And this is particularly prevalent in glioblastoma. And the reason I bring this up is because in glioblastoma, patients don't typically die from a metastasis. Almost every single patient that's diagnosed with the glioblastoma ultimately succumbs to the glioblastoma. And in patients that have surgical resection where we remove the glioblastoma prior to initiating treatment, 95% of those patients will relapse within 2 centimeters of this resection site, meaning that there's almost always small amounts here of residual tumor cells that we're unable to remove during the resection. And as we know, the brain is a very sensitive organ. So, we have to really do minimal damage to that organ as we're trying to remove the tumor. So how do we combat that the tumors are recurring exactly at the site where we know we know they are, but we're unable to remove them? And so I don't think I need to convince most people here that GBM is a difficult disease, but one of the most shocking statistics about this disease is that the standard of care has a median progression-free survival of only about 7 months, and the standard of care has remained unchanged for 20 years. We have not had a single new drug approved in glioblastoma in frontline in 20 years. We've had some medical devices, we've had some things approved for later line therapies, but in frontline glioblastoma, we're still doing surgical resection, chemo and radiation, and then hoping for the best when we know that these are what the outcomes are. The overall survival, the median overall survival is only 14 to 16 months, and there are about 300,000 new cases annually. So we're seeing this cancer just continue to be aggressive and really truly devastating. So our thought was, how do we use gamma delta T cells in frontline GBM treatment with the standard of care? How do we synergize with that standard of care to actually impact meaningful outcomes in these patients? And I just sort of set up for you why this is such a difficult tumor to tackle. So you're probably asking yourself, why would you pick this one? And so let me talk a little bit about why we picked this one. There are a lot of challenges to solid tumors, some of which I touched on, but some additional are actually making sure that your cells get to the right place. In solid tumor cell therapy, one of the issues is that when you give cells IV, a lot of them can go into sinks. They can sink into the lung, the liver, the kidneys, they can be cleared. You have to deal with all this first pass clearance, and whether or not they're actually migrating to the tumor site is a huge problem. And so if you don't know if your cells are getting there, then you don't know if they're effective. And so if your therapy doesn't work, your question may be, did the cells get where they needed to go? Yes or no? And if they did get there, then can you tell if they were working? So one of the things that we did is we tried to avoid that, and we can actually deliver directly into the brain. So we actually put a catheter into our patients, and we deliver our gamma delta T cells directly into the tumor bed during our treatments. The other side of this is that the brain has traditionally been considered immune privileged, and that is a plus and a minus. It's been challenging for many treatments in glioblastoma because getting any treatment across the blood-brain barrier is incredibly difficult. And this applies to immune cells as well. So there's very little immune cell trafficking across the blood-brain barrier. So it's very hard to get a cell therapy in. However, that means if we put our cell therapy into the brain, it's very likely to stay there and hopefully act in the way that we would like it to. The most important part of what I'm going to tell you here too is that the standard of care treatment for glioblastoma actually involves the use of TMZ or Temazar or Temodar or temozolomide. That's the chemotherapy that's given in standard of care GBM treatment. We know that TMZ is a lymphodepleting agent. And so when you give a cell therapy, we need to lymphodeplete so that your cell therapy can function in the way that you need it to. In frontline glioblastoma, we don't have to change the standard of care. We can actually use the temozolomide as our lymphodepleting agent. And the reason I'm going through all of this is because I think what's become really apparent in cell therapy is we really need to spend time thinking through our cell type and thinking through the indication. It's very easy for everyone to sort of follow one after the other and start doing the same indications and putting cell therapies in there, but thinking about why do we choose it? We're not choosing GBM because we think it's going to be the easiest. In fact, it's probably going to be one of the hardest. But does the biology make sense? Does the disease progression make sense? Is there a way you can dose that makes sense? And does the method of action of your cell make sense in this setting? So I think this is a really important point to spend time on before you push into the clinic with any cell therapy, is to think through, why are we doing what we're doing and how do we set ourselves up to eliminate as many variables as possible so that we have the greatest chance of being successful. So I'm going to tell you a little bit now about our program using what we call our Deltex DRI, which is drug-resistant immunotherapy, and I'll explain what that means. This program is called INB 200. So I spent a long time telling you that we wanted to leverage the power of the gamma delta T cell, but what does that actually mean? So gamma delta T cells, I told you, can naturally recognize cellular stress. We know that one of the most effective ways to stress a cell is to expose a cancer cell to chemotherapy. When you expose a cancer cell to chemotherapy, they start upregulating cellular stress signals, and then the which will essentially light them up like a Christmas tree or like a honing beacon for a gamma delta T cell to be able to recognize them and kill them. And so I'm showing you on the right here that we can actually increase cellular stress in chemotherapy-resistant glioblastoma. So the chemotherapy may not kill them, but it will still transiently upregulate stress, like ULBP2, BP4, 1, 3, MIC-A, and MIC-B. And on the bottom there, I'm actually showing you that we can also upregulate stress signals in the glioma stem-like cells. And so these are essentially the cancer stem cells in the GBM. So we know that we can upregulate cellular stress. The problem with this is that chemotherapy not only stresses cancer cells, chemotherapy will lymphodeplete, like I told you. So it will actually destroy your T cells. So we needed to figure out how to overcome this problem. So we genetically modify our gamma delta T cells, but we don't actually put a CAR on them. What we do is we actually identified the gene that makes chemo-resistant glioblastoma cells chemo-resistant. We hijacked it and cloned it into our gamma delta T cells so we can make chemotherapy-resistant gamma delta T cells. It's overexpression of a gene called MGMT. It's a DNA repair protein. And by overexpressing that in our gamma delta T cells, they can actually survive super therapeutic doses of chemotherapy. And we demonstrated all of this preclinically in a very nice publication that I'm happy to share. So I'm not going to go over the animal data today. I'm really just going to take a look at the human data. And so that's how we make our cells chemo-resistant. I also talked about synergizing with standard of care, and this is how we do this practically. So in the frontline chemotherapy, the frontline glioblastoma treatment, above the blue line here, you'll see exactly what we do in frontline. Patients are diagnosed, they very quickly go to surgery, they get surgical resection, and they immediately start chemotherapy and radiation daily. Once they finish that initial 6-week course of chemotherapy radiation, they'll have a break, and then they go into maintenance chemotherapy. And that maintenance chemo is usually 6 months, where they get dosed every 28 days. So what we decided to do is to go into the frontline therapy and layer our cell therapy right on top of this standard of care, because we want to leverage the stress signals that are caused by the chemotherapy to be able to paint those tumor cells with a target for our gamma delta T cells. So everything below the blue line here is what we do clinically, where we add our gamma delta T cells into this standard of care. So we dose every 28 days. We infuse our genetically modified gamma delta T cells that are chemotherapy resistant directly into the brain. And the whole idea behind this treatment paradigm that we've put together is that we know there are residual cancer cells there. We know that that's where the patient is likely going to relapse. And what we are trying to do by repeat dosing our gamma delta T cells is we are trying to use these gamma delta T cells every 28 days. We stress the tumor cells, light them up, use our gamma delta T cells to try to eliminate them. And if we can either hold the cells down so that they can't recur, causing a relapse, or eventually outrun them, we can hold these patients in remission or prevent these patients from progressing for as long as possible. So it sounds like a complicated treatment regimen, but again, thinking about how do we synergize in the upfront treatment setting so that we can get these tumors, attack these tumors with everything in our arsenal and give these patients meaningful time without progression. So this is what our clinical study looks like. Our patients started with either 1 dose, 3 doses, or eventually 6 doses of our gamma delta T cells, with the last cohort getting 6 doses. We manufacture our cell therapy between the surgical resection and the initiation of their chemoradiation. This is an autologous trial, and I'm going to talk a little bit at the very end, if I have a minute, about what are our thoughts about how do we look at the allogeneic market. And then we're looking at safety, MTD, time to progression, and overall survival. So I'm just showing you a very brief slide here of the demographics of these patients. And the purpose of this slide is that the median age is 66. These are older patients. They don't tend to do well. 54% unmethylated, so they tend to be less responsive to chemo. And 46% of these patients were a subtotal resection, meaning we know that there was some tumor that was visible that was left behind, which often happens because of the position of the tumor in the brain. The surgeons will only take out what is safe. So subtotal resections tend to do poorly. So we believe that we're seeing extended progression-free survival, and I'm going to show you that data right now. So this is a data cut that we presented at TCT of this past year. And so what I'm showing you here is we actually have the historical median progression-free survival that I told you earlier is about 7 months, and the historical median OS, which is about 14 to 15 months. In cohort 1, which is in blue here, they only got 1 dose. Eventually, all of these patients progressed and all of them passed. And we saw a slight improvement in median PFS, but not really significant at 8.3 months with a single dose. But when we started looking at repeat dosing, so 3 and 6 dose patients, in that instance, we have now seen a median progression-free survival of 16.1 months. We have more than doubled the median progression-free survival that you would expect based on standard care alone, and we've actually exceeded the median overall survival with progression-free survival. So this was extremely exciting to us because it really does, we think, demonstrate the power of this cell type and its ability, particularly with the repeat dosing, to keep these cancers and hold these patients without allowing them to progress. And so this is really the first time that we've seen such significantly improved median progression-free survival in the frontline setting. So we are continuing to follow these patients. We have not yet hit median OS. These patients are still going. We're looking forward to presenting a data update later this year. But this is sort of an example of how we are working to strategize how to use our cell therapy, leveraging what we think is intelligent engineering, and then putting them in the upfront setting in a complex treatment-resistant disease to try to synergize and to really drive meaningful outcomes in these patients. So here I just want to show you a little bit of biology, which we think is quite exciting. So I told you that these cancers tend to be very treatment resistant. They are in an immune privileged site. There's almost no immune cells present in the brain. So these tumors are typically very cold. And so here you can see on the left, this is a biopsy at diagnosis. You can see there's a lot of tumor tissue. That's the H&E stain. There's almost no infiltrating immune cells there. On the right, we actually have one patient that did eventually relapse after 6 doses. We went in and we actually biopsied this patient. And we saw in that biopsy that most of what we took out is actually dead necrotic tissue. And we see that there's significant immune cell infiltration. And the reason that that's so striking is because these patients have been getting temozolomide or chemotherapy for almost a year. Their peripheral immune cell counts and T cell counts are almost zero. But here we're seeing active and infiltrating gamma delta T cells in that tumor site. So that tells us that these cells are likely chemoresistant because they've been able to survive. But it also shows that they're continuing to act. And so what this suggests to us is that we would like to actually, in our phase 2, continue dosing these patients for as long as they're on maintenance temozolomide. So in some cases, if patients respond well, it can go up to 12 different doses. So we can keep dosing to keep driving down this tumor. And here I'm just showing you that we're also seeing at relapse infiltrating CD3 and CD8 T cells. So what we actually believe we're seeing is amplification and immune cell recruitment and activation of these other immune cell subtypes at the tumor site when we give our gamma delta T cells. And so the reason I bring this up is because we talk about allo versus auto. And this is exciting to us because this is an autologous product. An autologous product and an allogeneic product will have different release criteria per the FDA. So allogeneic typically having to be much, much more pure. So one of the questions we have here is, when we're seeing this patient benefit with the autologous product, will we see the same benefit with the allogeneic product? The autologous product does have some of these what we'll call carry-on cells. So it'll have some carryover cells. There can be some NK cells in there. There can be a small population of alpha betas in there. It's very pure gamma delta T cell, but not 100%. So is that providing some sort of benefit? And if we switched over to allogeneic, would we still see that benefit? And that's a question that we want to ask ourselves in the phase 2 study. So we actually have a design where we're going to look at autologous versus allogeneic cell products infused into the brain and try to answer that question. Because again, these patients tend to be older. Autologous manufacturing can be challenging in those populations. And some patients, though, for allo may not have a good donor. So, we want to see what differences we may see, and if both are successful or if we see a benefit of one over the other. And so really, we've seen a differentiated mechanism of action with gamma delta T cells. We've seen that they're linked to better survival, and we've also seen that that persistence has been noted in our therapies. We've seen enhanced quality of life, and we've seen long-term remissions. And with that, I will pause and happy to take any questions in the few minutes that we have left.
Thank you, Kate. It's a very really cool story and really inspired me as a former cancer biologist, study one of the hardest cancer. Yeah, I was gonna ask you why you guys choose GBM in the before this talk. Now I understand a little bit more. Yeah,
We get that question a lot.
no, it's a really tough challenge, right? So,
Mhm,
yeah, really exciting to learn more. How did you design your phase 2 to sort of elucidate
mhm.
difference between the auto and allo? I didn't realize I understand the gamma delta T cell are harder to manufacture, but I didn't understand there's other cell type maybe potential helping, is that who are you indicating a little bit as well? That
Yeah, I mean, and so this is something that people don't talk about in cell therapy manufacturing very often. So
Mm,
any autologous product is usually going to have some other, what we would call contaminating cell types, but because it's autologous, it's not generally thought to elicit any sort of immune response.
mm,
So many autologous therapies may have some percentage of other cell type in there, but since you're getting back your own cells, you don't have the same concerns that you would with an allogeneic therapy where you could potentiate some sort of immune response because gamma delta T cells can go from donor to recipient with no modifications, but things like alpha betas obviously cannot. Those may cause immune responses. So the release criteria for every company are likely a little bit different. But
mm,
in the autologous setting, you may not have a fully pure product in many cases. So one of the ways that we do this is so going into the phase two, we would start with an autologous therapy. We would immediately, and we've actually started that phase two trial. So that's been announced. We do have some patients that are enrolled and treated. So in that phase two, we can start immediately with the autologous. And then with the allogeneic, we would have to do a small safety run-in study. So like a phase 1B, which would be a handful of patients to demonstrate the safety of allo. I will comment that our leukemia study is an allogeneic product. So we
mm,
have demonstrated giving large systemic doses of allogeneic gamma delta T cells and the safety with that. But we'd still do a small run-in, phase 1B, and then we would move that into the full phase two. So, you do have to sort of quote unquote do the safety run-in.
mm,
You do have to submit your CMC to the agency, and that CMC may be slightly different based on whether it's allo or auto.
mm.
But that's all sort of the negotiation and the discussion with FDA.
Awesome. We do have another question before I let Kate go. Can you just touch a little bit about how you prevent gamma delta off targeting since you're leveraging cell stress response? Is there a
So
risk to, yeah.
that's a really good question. And so the answer actually is a lot simpler than than a lot of people want it to be. So gamma delta T cells in your own body are very, very sensitive at detecting healthy versus sick. So I talked a lot about how they recognize stress signals. They also have a whole set of receptors, and this is less well understood, but better in the last couple of years. A lot of research coming out from different groups on this. They have a whole set of receptors based on normality sensing. And so when they essentially will bind to a cell, they will sense stress and they will also sense normality. And based on that sort of seesaw, they decide whether or not they should kill that target cell.
Mhm,
So this mechanism we know is extremely sensitive in our own bodies. And what our hypothesis had been is that we could drive the cellular stress on cancer cells. And there are so many different types of cellular stress happening in those cells, not just the chemotherapy-induced DNA damage,
mhm,
but also things like the lipid membrane stress that starts becoming generated from rapidly dividing cells, that the gamma delta T cells would naturally be able to differentiate between cancer and healthy. And some of that actually comes from literature that shows if you do a gamma delta T cell knockout in mice, they spontaneously start generating tumors. So we know that the gamma delta T cells actually do immune surveillance in our own bodies to identify early oncogenic cells and eliminate them, but they don't eliminate healthy tissue. So we actually did a very large series of mouse studies demonstrating that even when we gave large doses of chemotherapy and infused these cells into the mouse brain, that we did not see any damage to healthy astrocytes, healthy brain tissue. We didn't see any damage to anything neurological or neural function in those mice. So we actually did quite a lot of data around that. And the agency accepted that, given that there were a number of other infused gamma delta T cell therapies that were kind of coming up, and they all were looking at the same safety data in different ways. So we were actually the first genetically modified gamma delta T cell to be put into a human. And I can tell you when we dosed our first patient, we were all waiting to make sure that that data held out. And we've seen to date no ICANS, no neurotox, no cytokine release syndrome, no negative impacts or treatment-related SAEs to date in any of these patients that we've treated, even those getting 6 doses in the brain. So it's a great instance of the biology bearing out when we moved
mhm.
into the clinic.
Awesome. Yeah, I know we're running out of time, but
Mhm.
that's kind of like tied with my question about what's so unique about those T cell that just chemo-resistant. But yeah, thank you, Kay and Neil,
Mhm.
for we're gonna conclude this webinar. I really appreciate both our speaker and all the attendees to participate today. Hopefully see you next time. Have a good rest of your day.
Great. Thank you so much.
Thank you.